Hi, I'm Faheem Seedat, a postdoctoral researcher at the University of Oxford and board-certified endocrinologist. Today, I'd like to share some highlights from our new study in Nature Communications, where we ask the question, how does the human pancreas adapt to meet the demands of pregnancy? During pregnancy, the body becomes more resistant to insulin, so more nutrients can reach the baby. To maintain blood sugar levels, the pancreas, and specifically the islet cells that produce insulin, have to adapt. Although animal studies have described islet adaptations in pregnancy, we know surprisingly little about what happens in humans. We addressed this question using rare, high-quality pancreatic tissue from pregnant and non-pregnant women made available to us by the Network for Pancreatic Organ Donors with Diabetes. We used three main techniques in our study: microscopy to isolate islet tissue from pancreatic tissue, and mass spectrometry to identify and measure thousands of proteins within the islets. In addition, we used tissue staining and advanced microscopic imaging to see specific cell types and molecular markers within the islets. Finally, we used a computer-based analysis pipeline to quantify changes in islet size, structure, and marker expression. In our study, we identified over 7,500 proteins within the islets, the largest dataset from human islets in pregnancy, and found that overall, protein composition didn't change much when comparing islets isolated from pregnant women to non-pregnant controls. However, there were some key differences that we noted in islets isolated from pregnant women. Islet area nearly doubled, the prolactin receptor increased in alpha but not beta cells, and the serotonin 2B receptor was absent in beta cells. Interestingly, GLP-1, a hormone that helps manage blood sugar, was significantly higher in alpha cells during human pregnancy. So why does this matter? Well, our findings show that human islet adaptation during pregnancy is distinct from the mechanisms described using animal models, which are the basis for most research today. To better understand and treat conditions like gestational diabetes, we need to study real human biology, and this study is a step in that direction. We're incredibly grateful to NPOD, the JDRF, and the Wellcome Trust for supporting this work. Thanks so much for watching.